In my presentation today, I will introduce the MxP Quant 1000 kit and the technology to you. So I will start with an overview about the key features. What are the key features of Quant 1000 and the coverage? And then I dive a bit deeper into the technology and the kit, and will show you and explain what are the components, what is the workflow, and also the timeline. So in my overview part, I will start with the key features and why Quant 1000 makes the difference. What is the coverage, and also what is the Quant 1000 family? So it's not only the Quant 1000 kit itself. So metabolomics is a very powerful tool to understand chronic disease, as you learned from Alice. Because chronic disease is deeply influenced by lifestyle and exposure that are not measured or not visible in the genome. So researchers want to solve health problems. And from that work, we expanded our panel with small molecules highly relevant to chronic disease. So what are the exciting key features of Quant 1000? With 1,233 metabolites across 49 classes, we have significantly expanded our metabolite portfolio with small molecules. The kit has been validated with human plasma and urine and requires a sample volume of only 40 microliters. But we go beyond just measuring metabolites. On top of the actual metabolite panel, you get 648 predefined metabolite sums and ratios with biological or clinical relevance based on published scientific research. What facilitates comparisons across different groups or patient cohorts and improves biological interpretation. And this coverage of metabolites and its sums and ratios results in 1,881 biomarkers, as every measurement can be a metabolite or a ratio or a sum of metabolites is a potential biomarker. And with this, we are reaching a new era in targeted quantitative metabolomics. And this enables a holistic view of metabolic health and disease and empowers researchers to explore hundreds of metabolic pathways. So whether you're working on biomarker discovery, disease research, or fundamental metabolism research, the kit ensures you get the most reliable and actionable metabolomics data available today. That's the difference we bring to research. So let's take a look on the metabolite classes and numbers. So here on the left, you find the small molecules. On the right, the lipid panel. Compared to our previous MxP Quant 500 and Quant 500 XL kits, we have dramatically expanded the small molecule panel from about 100 across 14 classes to now 327 metabolites across 24 classes in Quant 1000. The classes here are highlighted in yellow were expanded with more metabolites compared to Quant 500. The green ones are new biochemical classes. So let me pick a few. We have increased a lot on the classes that feed into the TCA cycle or are intermediates, such as fatty acids or carboxylic acids or the new class of dicarboxylic acids. So with this kit, we now provide nearly complete TCA cycle coverage. And this is really exciting as this offers new insights into mitochondrial function, energy metabolism, and neurodegeneration as Alzheimer's disease, for example, as you learned from Alice. Another example, the kit now includes a dedicated panel of polyamines that enables researchers to study their role in tumor metabolism and potential therapeutic targets. This addition makes the applicability of the kit in oncology much stronger and supports biomarker discovery and cancer metabolism research. The lipid panel on the right side is similar to our previous products and covers 906 lipids across 25 classes. When you see all these incredible numbers, you might be wondering, how does this compare to untargeted approaches? So please allow me to elaborate on this. Untargeted solutions can screen and identify several thousands of metabolic features, but untargeted data contains many features with unknown IDs that might or might not be regularly present, leading to a maximum number of identified compounds that is often around the same number of metabolites quantified by comprehensive targeted solutions. In other words, the comprehensive panel of Quant 1000 can offer actionable, equivalent, or even higher coverage that is usually reported by untargeted methods. And this is our value proposition. The targeted quantitative method of Quant 1000 gives higher level of information by focusing on defined metabolites relevant to biochemical pathways. So now let's come to the Quant 1000 family. On the left side, you see the complete solution with the complete panels of small molecules and lipids. The small molecule panel and lipid panel of Quant 1000 are also available as separate kits. So if the focus is on small molecules only, you can have this part as MX Quant kit. For example, who want to study the effects of microbiome on the host, or more generally explore core metabolism. If lipid metabolism is of primary focus, this part is available as Alex Quant kit, which has a broad panel of circulating lipids and membrane lipids from lipoproteins. Each panel, small molecules and lipids, has its own refrigeration box here in ice blue with the kit plates, calibrators, and QCs, and so on, and a non-refrigeration box in dark blue with consumables like collection plates and coverings. So over the next slides, I would like to dive deeper into the technology and the kit design, starting with an overview. And then I have a slide showing the detectability and analytical performance. Then I will show you which components are included and how does the plate design and the lab workflow look like. So let me give you some general technical information first. So for Quant 1000, you need 40 microliter, as mentioned, of plasma or urine, or 200 milligrams of feces wet weight maximum, actually. So actually you need less, but less is often hard to handle. So we recommend 200 milligrams. The kit has been validated for human plasma and urine and tested for human feces and other matrices like animal plasmas and mouse liver tissue and milk samples. The results across 9 matrices in total we tested were published in a poster presentation at ASMS, and I have a summary for you on the next slide. The supported instruments today are the Agilent 6495C, the SAIX 5500 Plus and 6500 Plus, and the Waters Xevo TQ-XS. But we are not sleeping, and further platforms will follow until the end of the year. Regarding throughput, you can run up to 80 samples per kit. If you have 1 instrument available, you can run up to 2 kits per week. If you have 2 instruments, you can run up to 3 kits per week. And the total time per kit, so from sample, from freezer actually to results, is about 69 hours. But I will have a separate slide on this later. The expected detectability, what means above the limit of detection in more than 50% of samples, is as follows. So we could find over 1,000 metabolites in human plasma, over 500 metabolites in human urine, and over 910 metabolites in human feces. So these results are published in a tech note already and are summarized on this slide, along with other matrices that we recently tested. So on this slide, I want to give you an overview of the detectability in common sample types. For the animal samples, we tested 5 individual samples, and for the human samples was 10 or 11. 2 interesting findings here. The detectability is comparable in plasma across human, rat, and dog, all with over 1,000 detected metabolites in at least 50% of the samples that were tested. And despite of many lipids not present in urine samples, we could still find more than 500 metabolites in more than 50% of human urine samples. Important to mention here, these numbers only serve as orientation. So we only tested a small number of samples to get an idea about the detectability. And the detectability strongly depends on the nature and the quality of the samples, and also on the instrument condition, of course. And on this slide, I want to give you some insights into the analytical performance and reproducibility in particular of the kit across different sample types and different mass spectrometers. So in the graphics here in the plot, the left red dotted line is at 15% CV, what is the coefficient of variation, and the right dotted line at 25% CV. So we found that the majority of analytes were well within or below 15% CV, and the average CV was in the one-digit range, below 10% across all samples and across all instruments. The results about this performance across MS platforms will be published next week through our monthly newsletter in a technical note, also including accuracy plots against the NIST standard and correlation plots demonstrating the comparability across mass spectrometer platforms. So if you're interested, please visit our website and sign up for the newsletter. So now let's talk about the kit components. So what customers value from biocrates technology are the ready-to-use components and methods. And this we also deliver with the MxP Quant 1000 kit. So our users will get kit plates with integrated internal standards, lyophilized test mixes, calibration standards, and quality controls, instrument-specific methods, which are already optimized and validated. And there is also an analytical column, a pre-column available separately for the small molecule panel for the analysis. Regarding documentation and support, you will get a quick start guide and a full user manual with all these detailed instructions. We also have protocols for different matrices. So the user manual includes human plasma, but we also have protocols for urine, for tissue samples, for fecal samples, for example. We provide a list of required lab equipment and chemicals that is required to process the kit in the lab. So for example, a nitrogen evaporator, or alternatively a pressure manifold, plate shaker, pipettes, and so on. And what you also receive from us is technical support and the training by our biocrates specialists. So our technology is standardized and easy to use and also automated. And I will elaborate later what automated means. In addition to the kits, we also provide the analytical column and pre-column that is available separately to the kits and required to analyze the small molecule panel. So it's a column that is good for approximately 9 to 10 kits, depending on the type and quality of samples, of course, and the pre-column. You can also order a column package consisting of 1 column and 3 pre-columns. And the good thing here is that this setup requires no further holder for the pre-column. So this kit-specific column has been validated as part of kit development and is a must-have to ensure standardized performance, reliable chromatographic separation, and reproducible results. On this slide, I would like to show you how does our kit technology actually look like and why we have built it like this. So here you see a cross-section of the kit plate, which has a patented design. The filter plate of the kit is spiked with additional holders with FDA-approved filters where internal standards are already incorporated. And this design allows for simple and fast sample preparation, but also for efficient derivatization, extraction, and purification of the samples. And together with the lyophilized calibrators and QCs and test mixes, this is the hardware of Quant 1000. Now let's talk about the heart of the kit, or the 3 hearts actually. Quant 1000 consists of 3 kit plates, 2 for small molecules and 1 for lipid analysis. I would like to start with the lipids on the right side. So the lipid panel and coverage is almost identical to our previous Quant 500 XL kit, where they were distributed over 2 plates and also combined with small molecules. What is new in Quant 1000 is that all lipids are now combined on 1 plate for pure lipid analysis. And this allows us the modularity and to offer the lipid and small molecule panels separately. The lipid panel comes with a lipid plate with all internal standards already integrated, capture plate, 2 dilution plates, QCs, test mixes, and an additive to prepare the mobile phase for the FIA run. FIA means flow injection analysis, so no analytical column is required for the lipids. what has a couple of benefits. What are fast run times on the one hand, and also high lipid coverage on the other hand. So one injection is only 3 minutes, and the over 900 lipids are analyzed by 4 injections, 3 in positive and 1 in negative mode. Now let's talk about the really new thing, the small molecule panel. So due to the complexity of the metabolome, there is no one-fits-all solution or method. So we broke down the metabolite panel in 2 substance groups using selective reagents for derivatization. One is 3-NPH, what is 3-nitrophenylhydrazine, that reacts with the carbonyl and carboxyl groups. And the other one is PITC, what is phenyl isothiocyanate, that reacts with primary and secondary amines. Based on this, it was defined, of course, which analytes go into which plate. The analytes that are not derivatized and were tested on both and finally added to the plates where they perform better. A PITC derivatization we already have in our previous kits. So the 3-NPH derivatization is the new thing here. So what do we mean with enhanced performance? So let me give you 2 examples. The fatty acids that were analyzed on the PITC plate earlier, but not derivatized, are now analyzed with the NPH plate, which runs in the negative ion mode, and all the fatty acids are now derivatized. And that means better signals and more robust performance. And this also allowed us to add much more fatty acids into the kit compared to older kits. So now there are over 40 fatty acids covered. For the PITC plate, we are using now triethylamine as reagent instead of pyridine. This results in stronger signals and improves the detectability of low abundant metabolites. The sample preparation time and run time I will resolve a bit later on a separate slide. With this slide, I want to give you some insights into the workflow. So I picked one plate now, the NPH plate, as an example to give you an idea about the workflow in the laboratory. So, of course, it starts with thawing the samples and reconstituting the QCs, the calibrators, and the internal standard mix. You assemble the plate. That means you just attach the filter plate to the collection plate with tapes. Then you start adding your sample. Then you add the EDC mix and the 3-NPH mix, what is for derivatization. Then you incubate for an hour at 35 degrees. So using a plate heater, you can use a plate shaker with a heated lid for this, or just an oven, for example. Then formic acid is added to stop the derivatization reaction. This is incubated for 5 minutes. Then you dry the plate for 1 hour. Then you add the internal standard mix, and then you add an extraction solvent and you shake it for 30 minutes. And during this step, the proteins will participate and will be removed. And the last step, the filtration, is actually also the purification step. So you can use either a plate centrifuge for that, or a pressure manifold. And then you receive your extract down in the capture plate, and this is already ready to go. So you cover it with a silicon mat. You seal the plate. Silicon mat is also provided with the kit, and you place it in the autosampler and can run the kit. So that was the hardware part. But I cannot talk about the kit without talking about software. So Quant 1000 is available embedded into a total solution. So it's part of an ecosystem consisting of the kit itself, optimized and validated methods specific for key triple quadruple mass spectrometers, and the workflow manager WebIDQ, what is a cloud-based software, what I will show you now. WebIDQ is a kit companion that takes and guides the user through the entire workflow, from project management through metabolite quantification to results and even statistical analysis. So before preparing and running the samples, they are registered in WebIDQ. The plate layout can be customized, and the sample sequence is directly exported to the MS software and ready to go. after the measurement. And here are some insights into the software, just to give you a look and feel. The raw data is directly uploaded and processed, starting with AI-driven peak identification and integration, automated quantification, validation, where accuracies and CVs are checked, but also signal intensities. Data normalization can be performed, and the integrated metabolic indicator tool automatically calculates the predefined sums and ratios that I mentioned before. And you can transition seamlessly from quantification to statistical analysis within WebIDQ without switching software. So it's all in one platform. Here you see the total project timeline and a summary of the kit workflow. So it starts with the project design in WebIDQ and with the system preparation, where you run the test mixes and do the system suitability test. Then you prepare the 3 kit plates in the laboratory. Then you run the plates. So the small molecule panel for the LC part needs 32 hours measurement time over the 2 plates, and the FIA part for the lipids 22 hours. And then you do the quantification and validation in WebIDQ, so the whole data processing, and you can check the peaks and the calibration curves and the validation and the performance results and so on. And then finally, what you can also do in WebIDQ is a data cleaning and imputation, data normalization, using the metabolic indicator as just mentioned, doing statistical analysis, and finally export the results. So we are really proud of our new solution, because metabolomics is only as powerful as the technology behind it. And that's where MxP Quant 1000 makes the difference and solves key challenges. It sets a new standard of excellence in metabolomics profiling, because Quant 1000 delivers the most comprehensive quantitative measure of metabolism. And thanks to its high analytical precision, Quant 1000 delivers deep biological insights from just minimal sample volume, making it a powerful tool for fundamental, pharmaceutical, and clinical research. So MxP Quant 1000 gives scientists the solution to decipher chronic disease mechanisms and to identify phenotypic changes, contributing to the development of innovative solutions that are driving the transformation of medicine. So this extensive coverage, and this is my last slide now, ensures that whatever your focus is, you have the right tool to uncover meaningful insights. In fundamental research, scientists aim for biomarker discovery or need comprehensive insights to understand the biological mechanisms underlying chronic disease or chronic inflammation in people living with HIV, what we will see in a minute from Chris. If the focus is on systems biology approaches, integrating multi-omics with genomics, transcriptomics, and proteomics, with Quant 1000, you can do that. In clinical research or pharmaceutical industry, early-stage biomarkers are a key focus area for understanding disease mechanisms and identifying potential therapeutic targets. Customers today or consumers today are more proactive about their health than ever, seeking ways to monitor metabolic health before disease develops. While consumers don't use our products directly, routine labs and healthcare companies rely on our products to bring them reliable screening solutions to detect metabolic imbalances and help consumers take preventive action. So once more, with Quant 1000, you can do that. So no matter what your research focus is, Quant 1000 provides the precision, the coverage, and the insights you need to make a difference. And with this, I would like to close my presentation, and thank you for your attention.